Hello, my name is Abby Hermanson, and I will be talking about the effects of troponin on tyrosinase kinetics. Now, just some background information on tyrosinase. It is involved in the pigmentation processes and the browning effects that is caused in many living organisms. And this is due to the bicopper active center that can bind two oxygen molecules. And this results in four oxidative states. There's the deoxy, oxy, met, and deact tyrosinase, which each all have their unique kinetic properties. And you can see here in this image the way they interact with each other. It also has different isoforms that are very tissue specific and depend on the oxidative states that were mentioned and genetic information and other environmental factors. It also has two forms, so it's active and latent form. The active form usually occurs during a neutral pH and has a subunit of about 43 to 48 kilodaltons. Its latent form usually occurs at lower pHs, about like 5.5, and has a subunit of 62 kilodaltons. And it usually gets inactivated during the monooxygenase transformation of catechol, which causes one of the copper ions in the active site to be removed. And so it gets converted from the oxy to the deact form. And this operates usually under two catalytic functions. There's the monooxygenase mechanism and the oxidase mechanism. So in this image, this is the browning mechanism. You can see how tyrosine gets converted into melanin and how important tyrosinase is throughout the entire mechanism. In the experiment, the white button mushroom was used, and in the process, it is very important to the mushroom that tyrosinase is there as a defense against free radicals and UV rays, gamma radiation, dehydration, and extreme temperatures. So tyrosinase plays a huge role in the white button mushroom. It is also important to note that there is a lot of similarities to tyrosinase enzyme in humans as well as found in the mushroom. So this implies that tyrosinase can be isolated and purified and be used for perhaps pharmaceutical treatments for melanin issues in humans. And so this study is to see the kinetic enzyme activity of tyrosinase from the white button mushroom with and without an inhibitor, which we'll be using troponin, to aid in treatment designs for skin pigmentation disorders in humans. Now, since troponin will be the inhibitor, some background on that. It is a slow binding inhibitor. It has mixed inhibitory effects. It is one of the most potent inhibitors and has 50% inhibition in tyrosinase. It also inhibits both the mono and O-dihydroxyphenolase activity of tyrosinase. And data suggests that the competition between troponin and the O-dihydroxyphenols for the copper ion occurs in the active site. And it only binds to the oxy isoform of tyrosinase based off of the incubation studies. Increasing troponin concentration causes the initial velocity of the final steady state rate to decrease as the product accumulates in the progressive curve, meaning more troponin, less product forms. So this is the results. This is the first table is the tyrosinase kinetics data, and pretty much this shows the accuracy of our data during the activity of it. And this is varying amounts of catechol every 10 seconds for 1 minute. All the samples contained 4 microliters of tyrosinase, 20 microliters of SDS, and the rest was filled with 100 millimolar sodium phosphate buffer at pH 7 to fill up the 2000 microliter volume. And so in Series 1, the varied amount of catechol was 400 microliters, in Series 2 was 200, in Series 3 it was 60, and in 4 it was 20. Now, as you can see, we got pretty consistent results, except it was expected that Series 1 would have a higher activity than the rest, seeing as more substrate concentration being the catechol, the more activity would occur as the substrate binds to it. However, that is not the case in this experiment. Now, this one is very similar to the one before, except now the inhibitor tropolone is introduced. So the figure, same thing, is recorded every 10 seconds for 1 minute. In this case, it is 5 microliters of tyrosinase, mainly because it is better to have more concentration, more activity, and I joined another group, so that's also another reason. And 20 microliters of SDS. The rest was filled with the 100 millimolar sodium phosphate buffer, pH 7, to fill up the volume. It is also important to note that it was also varying amounts of catechol during this. The series 1 had no tropolone as a sort of basis. So series 2 through 4 had 10 microliters of tropolone. So series 1 had 120 microliters of catechol. Series 2 had 200 microliters of catechol. Series 3 had 100 microliters of catechol, and Series 4 had only 40 microliters of catechol. So it was expected that Series 2 would be the highest. As you can see, it is one of the lower ones, just based off the concentration of the substrate being greater at that point. So this was not what was expected. So this is table 1. This indicates the catechol concentration of a twofold serial dilution and the relative activity of the data of each trial, with and without tropolone. So the trials 1A through 8A is without tropolone, and trials 1B through 8B is with the tropolone. So you can see as the concentration gets cut in half from every single trial, and you can see on the third column the relative activity. And this was not the results we expected. We expected that the higher concentration of catechol would have a higher relative activity. And as you can see from 1A to 2A, that it actually went up during the dilution, and from 3B on, it was negative numbers, which you're not supposed to have negative activities, so our results were a bit odd. This is the Eddy-Hofstee plot of tyrosinase. This is without the inhibitor. Each trial contained 4 microliters of tyrosinase, 20 microliters of SDS, and the rest was also filled with the 100 micromolarity sodium phosphate buffer pH 7. And this corresponds with the dilution sample of catechol based off of the table in the slide before. So trial 1A had catechol concentration of 16, trial 2 had 8, trial 3 had 4, 5 had 2 millimolarity, trial 6 had 1 millimolarity, and it just kept getting half by there. The R square value, it is important to note, is 0.0319, which is not great, seeing as we want to get closer to 1. So our points were a little scattered. This one is kind of worse than the one before it. This is with the tropolone inhibitor. And this had 5 microliters of tyrosinase, 10 microliters of tropolone, and that was consistent throughout it. 20 microliters of SDS, and the rest was filled with the same 100 micromolar sodium phosphate buffer pH 7. And concentration of catechol was decreased by half per trial. And it is important to note that the R-squared value was 0.5753, which seems better, but I'll talk about the table in just a moment, but the Ki value was undefined just based off the data. The main reason being we got negative values for the Km, and the data should be in the first quadrant, not in the third. So just a little thing to go off of. This is how it should look like for a mixed inhibitor, which tyrosinase is. And this is our data based off that. So the Vmax was, without the inhibitor, was 0.1248. With the inhibitor was 9.26. And there's our range, the Km values. Now, as you can see, with the troponin, we got a negative Km value, which should not happen. It typically, Km values, the smaller it is, the more higher affinity the enzyme has for the substrate. And we expected that there would be a higher Km for with troponin because the tyrosinase wouldn't be able to bind to catechol as well. But it is a negative value, which should not be that. And then the Vmax Km value is determined by, if I go back, by where it intersects on the y or the x-intercept. And in this plot with tropolone, it doesn't intersect the x-axis. So that is also another reason why Ki is undefined. So based on the data from figure 4b, the inhibition mechanism cannot be determined. However, research has shown that tropolone is a mixed inhibition mechanism. The inhibitory effects of tropolone for the white button mushroom with L-DOPA in other research papers has shown that the substrate to have a Ki value of 5 to 40 nanometers. And the Km value of tyrosinase without tropolone was 0.37719. With tropolone was a negative value. However, Km cannot be negative, just like I mentioned. The Km value from the research article explained with L-DOPA was approximately 0.46 millimolarity, which is higher in what was expected than without the inhibitor. So in future experiments, how to fix this, I believe the main issue was our concentration of tyrosinase in the sample wasn't high enough to register the activity well enough. It might have if we had also given it more time to react with the substrate, but in any case, I believe increasing the amount of tyrosinase would help with the experiment. Here are some references that were used, and the numbers are throughout the presentation to refer back to. But thank you very much, and that is my presentation.